Hello friends, today we are going to discuss the topic chemistry of carbohydrates. This is one of the important topics of biochemistry. So let's begin. Carbohydrates are widely distributed in plants and animals. In plants, glucose is synthesized from carbon dioxide and water by the process photosynthesis, and it is stored as starch or as cellulose, which is a part of the plant framework. Animals can synthesize carbohydrates from the various lipids, cholesterol, and amino acids. And most animal carbohydrate is mainly or ultimately derived from the plants. And carbohydrate has very important structural and metabolic role in our body. So definition. Definition of carbohydrate is carbohydrates are defined chemically as aldehyde or ketone derivatives of higher polyhydric alcohols or compounds which yield these on hydrolysis. So from the definition you need to understand they have a functional group which will be either an aldehyde or a ketone, and apart from that they are alcohols. So they are polyhydric alcohols. So they'll have multiple alcoholic groups in them. And examples are glucose, fructose, maltose, inulin, starch, glycogen, hyaluronic acid, heparin, chondroitin sulfate, etc. As we go ahead, you will come across all of them and you'll understand the complexity of these carbohydrates. After definition, let's proceed on to the functions of carbohydrates. Carbohydrates form the energy source of our body. D-glucose, which is also taken as the form of glucon D or the dextrose, is the most readily available source of energy. 1 gram of glucose gives us 4 kilocalories of energy, which is equivalent to 16 kilojoules of energy. And important to remember is that brain and RBCs, they are solely dependent upon glucose for energy. Coming on to the next function, structural framework. Carbohydrates form the most important structural framework of our body. Mucopolysaccharide form the ground substance of the mesenchymal tissue. Cranial cartilage is formed of chondroitin. Nucleus pulposus, wall of aorta, cornea formed of keratin sulfate. And as glycoproteins with collagen and elastin and oligosaccharides, they are all components of the carbohydrates. Importantly, ribose and deoxyribose, which form our RNA and DNA, are taken from the carbohydrates. And the structural elements of the cell wall of bacteria is carbohydrate. Continuing with the function, they are the constituents of various antibiotics, drugs, important ones like being digoxin, which is a cardiac glycoside, quabin, which is a sodium pump inhibitor in the cardiac muscle, and fluoridine, which is a sodium dependent transporter across the mucosal membrane. They are all carbohydrate moieties. Then amino sugars, they can also have constituents of carbohydrates like erythromycin, carbomycin. Then carbohydrates act as plasma expanders. A special type of carbohydrate, dextran, is a plasma expander. Then blood grouping substances, various neutral mucopolysaccharides contain galactose, fucose, glucosamine or galactosamine, and they are the blood grouping substances. Heparin, which is a heteropolysaccharide, is a natural anticoagulant. Then hydrolyzed starch, we can use it in form of mucilages and also in the form of formation of infant formula where it is easily digestible. Now, major diseases associated with carbohydrate metabolism include diabetes, galactosemia, glycogen storage disorder, lactose intolerance, and mucopolysaccharidosis. So only from this thing you can realize that carbohydrates is a very big topic and lot of emphasis and lot of revision is required to understand this topic clearly. Let's start with classifying carbohydrates. We use the word saccharides. Saccharides, what does it mean? It is a word which is used to describe a sugar. One sugar molecule is known as monosaccharide. Two sugar molecules as disaccharide. 3 to 9 sugar molecules as oligosaccharide, and more than 10 sugar molecules as polysaccharide. So that means carbohydrates are classified into monosaccharides, disaccharides, oligosaccharides, and polysaccharides. Coming on to monosaccharides. Monosaccharides are the simplest form of carbohydrates and they cannot be broken down or they cannot be hydrolyzed into more simpler carbohydrates. The empirical formula of this is CH2O n times, which literally means it's a hydrate of carbon. It is classified into two different categories depending upon the number of carbon atoms and on the number of the functional group, which can be aldehyde or ketone. So these are the different carbohydrates. The trioses means the simplest carbohydrate is a triose, then a tetrose. Triose has 3 carbons, tetrose 4 carbon, pentose is 5 carbon, hexose is 6 carbon, and heptose is 7 carbon. So they are classified depending on the number of carbon atoms. Now, once we know the number of carbon atom, we have to find out they'll have one of the functional group, which can be an aldehyde. If it is an aldehyde group, then the 3 carbon monosaccharide will become glycerose, 4 carbon will become erythrose, 5 carbon is ribose, and 6 carbon is glucose, 7 carbon is aldoheptose, right? But if the functional group is a ketone, then 3 carbon monosaccharide is dihydroxyacetone, 4 carbon is erythrulose, 5 carbon is ribulose, 6 carbon is fructose, and 7 carbon is pseudoheptulose. So it's like number of carbon atom and then what functional group they have. This classifies the monosaccharides. Now let's talk about some physiologically important. Now glucose is the most important monosaccharide in our body. It is the most readily available form of energy for our body. It is the chief physiological sugar which is present in the blood, and you already know that brain and RBCs depend exclusively on glucose for their energy needs. Most dietary carbohydrate is absorbed into the bloodstream as glucose. It occurs as a constant of disaccharides and polysaccharides. It is a universal fuel for brain and for the fetus, and it is stored in liver and muscle as glycogen. It shows mutarotation. We'll understand what mutarotation is when we read about the various properties of carbohydrates. So mutarotation, it can occur from left to right or positive to negative. So this shows a positive mutarotation which is stabilized as 52.5. The alpha form shows a mutarotation from 102 degrees, whereas the beta form is stable at 19 degrees. But when in a solution, they stabilize at plus 52.5. That means it is dextrorotatory solution. Glucose is a precursor for synthesis of all other carbohydrates. Glycogen for storage, ribose and deoxyribose, galactose, glycolipids, glycoproteins, and proteoglycans. As we go through the topic, we'll be talking about glucose and all its products in detail. But over here, I'm just giving you a glimpse of what all substances glucose forms. The next monosaccharide of importance is galactose. Galactose, it occurs in combination with other sugars in plants and animals. It is a constituent of milk sugar, which forms a disaccharide known as lactose. It is a constant of lactolipids and glycoproteins, and it is an epimer of glucose at C4. Now what do I mean by saying epimer? It's another isomeric form of glucose. So it differs from glucose at position 4 only. All the structure is completely similar except of the orientation of the hydroxyl group around the C4 carbon. It is less sweet than glucose and less soluble in water. It is also dextrorotatory and shows mutarotation. Again, it also shows the final mutarotation in positive direction, so it is dextrorotatory. Alpha form has a rotation of 150.7, whereas beta form has a rotation of 43. And when in solution, it stabilizes at the rotation of plus 80 degrees. The next important monosaccharide to be discussed is fructose. Now, fructose is a ketohexose, which is also known as commonly as fruit sugar. The first two carbohydrates that we discussed were the aldose sugar, means that they have the functional group as an aldehyde. Fructose is a ketose sugar and it has ketone as the functional group. It is a constant of sucrose, which we also know as table sugar, and also of the polysaccharide inulin, which is an important exogenous glomerulofiltration marker. Seminal fluid is rich in fructose and it acts as a source of energy for sperms. So in patients who have infertility, In males, levels of fructose are estimated to find out if they are the cause for immobility of the sperms. Now, according to the properties, fructose is levorotatory. That means it rotates the plane polarized light towards left, and it is also known as levulose. Now, in a solution, it stabilizes at a degree of minus 19 degrees, which is towards the left. The alpha form is stable at minus 21, whereas the beta form is stable at 133 degrees. Moving on to disaccharides. Disaccharides are the condensation product of two same or different molecules of monosaccharides. So the journal formula for disaccharides is CnH2ON-1. So this loss of water molecule occurs in the formation of glycosidic linkage. So two molecules of monosaccharides combine to form the glycosidic linkage with loss of one water molecule. The common examples of disaccharides are maltose, made up of two molecules of glucose, lactose, which is also known as milk sugar, made of glucose and galactose, and sucrose, which is made up of glucose and fructose. Other name for sucrose is the table sugar. Now let's see how the constitution occurs and where the linkage is. The sucrose, which is a disaccharide, also known as common table sugar, is made up of glucose and fructose. So the alpha 1 carbon of glucose is joined to the second carbon of fructose, and we call this linkage as alpha 1,2 linkage. So this is very important to remember because this is the linkage because of which sucrose becomes a non-reducing sugar, because glucose at position 1 and fructose at position 2 contain their functional groups. So we know that sucrose is non-reducing and it is not acted upon by the Benedict's solution. It will give a negative Benedict's test when we test for sucrose. The next is maltose, which is a product of starch hydrolysis. So whatever carbohydrates we eat in our body, they are hydrolyzed into maltose, or we can get maltose from other sources. The linkage in maltose is between two glucose molecules, and the carbon 1 of the first glucose and carbon 4 of the fourth glucose are linked, and the linkage in maltose is alpha 1,4 glycosidic linkage. The third important disaccharide is trehalose, which is found in a fungi, and the linkage over here is between two molecules of glucose and both at the first position of the glucose. So next is lactose, which is also known as the milk sugar, and it is formed by the combination of two monosaccharides, galactose and glucose. Galactose, the first position of galactose, which is in beta orientation, is joined with the fourth position of glucose to form lactose. Coming on to the biomedical importance of disaccharides. Hydrolysis of grains contain lots of maltose and it's used for preparation of various foods which is easily digestible, especially the baby food. Lactose present in breast milk is a good source of energy for the newborn. Lactose is fermented by E. coli to form lactic acid, and this also causes the souring of the milk. And parenteral infusion of sucrose is beneficial in cerebral edema. So coming on to oligosaccharides. Oligosaccharides are the condensation product of at least 3 to 10 monosaccharide molecules. Less than 3 are disaccharides and more than 10 are polysaccharides. They are present on the extracellular membrane surface, on which they are attached covalently as integral membrane protein, and they are rich in information and they have functional importance. They are also present on antibiotics, coagulation factors, immunoglobulins, peptide hormones with terminal sialic acid residue. They actually are the markers of passage of time, and they determine when a particular substance with which they are attached needs to be taken out of circulation. And when they are attached to proteins, they prolong the longevity of the proteins. Once they get detached, like if sialic acid is attached, so sialysis will occur. And once sialic acid is dissociated from a particular substance, a particular protein, so then hydrolysis of that protein takes place. So in a way, they are very important. They are integral membrane surface proteins and they contain all the information and they help in the receptor functioning also. Coming on to next, polysaccharides. Polysaccharides are when we have more than 10 monosaccharide units, and we also call them polyglycans. So more than 10 monosaccharide molecules will join to form polysaccharide molecules. Now, a thing to note over here is when we talk about carbohydrates, the polysaccharides, they do not have a fixed chain length. We cannot say like glycogen has 250 glucose molecules. It is never like that. The size of the glycogen molecule or any polysaccharide varies, whereas for proteins we have fixed length, a particular peptide length, like insulin has 51 amino acid, but for carbohydrate polysaccharides there is no fixed length. Now these polysaccharides have a general formula of C6H10O5N, and they're further classified into two different types. So they are classified into homopolysaccharides. That means they are made up of polysaccharide chain made up of the same monosaccharide unit. For example, we have a monosaccharide unit A. So it's A linked to each molecule through a glycosidic molecule. Another example is starch, which is made up of, which is a homopolysaccharide of glucose. So there are glucose molecules, they are linked together. Glycogen, another homopolysaccharide of glucose, dextran, another monosaccharide of glucose, cellulose, glucose, inulin, a polysaccharide of fructose, and dextrins, again a homopolysaccharide of glucose. Heteropolysaccharides, from the name indicates, they have two different, maybe three different monosaccharide units present in them. Okay, so as we go ahead, we will learn about all of these. So coming on to the structure. So homopolysaccharides can be branched or unbranched. So this is a chain. This is a 6 carbon molecule representing glucose. So this is an unbranched glucose polymer, and this is a branched glucose polymer. Homopolysaccharides can be branched or unbranched. For example, cellulose, which is unbranched. So how is it linked? It's linked, the glucose is in beta conformation, D form of glucose. Another example of unbranched homopolysaccharide is inulin, which is a polymer of fructose. When we talk about branched polymers of glucose, they are starch and glycogen. Coming on ahead to heteropolysaccharides or heteroglycans. Now heteropolysaccharides, they can be of two different monosaccharide molecules or maybe more than two. So they can be multiple, and both they can be unbranched and branched. Now heteropolysaccharides are composed of a mix of several types of monosaccharides or derivatives. Unbranched contain only two types of subunits, whereas the branched contain more than two types of monosaccharide units. They're present everywhere and perform high diverse functions. The most important ones being protection of the shape, maintaining the shape, the extracellular support for the various organs and tissues of the organism. Now heteropolysaccharides, we classify them broadly into two types. Now this is going to, you will have to revise this again and again because it's a little lengthy one and it takes time for you to understand. So mainly of two types. First broad categories of non-nitrogenous and nitrogenous. Non-nitrogenous means there is no sugar amines that are present. So under this you have plant gum mucilages, which is used as an emulsifying agent. You have pectins, gels, major component of the infant formula, and the agar, the one that we commonly use for the cultures in microbiology. The second broad category is the nitrogenous ones, which has amine sugars. So these amine sugars can be neutral or they can be acidic. The acidic content defines like how much of reactivity that compound will have, and the acidity is mainly because of the presence of the sulfate radicals in these molecules. So let's classify heteropolysaccharide. Heteropolysaccharides are classified into two main categories. They can be non-nitrogenous, means they don't have any amino sugar, or they can be nitrogenous. The non-nitrogenous group contains plant gum, mucilages, pectins, and agar, whereas the nitrogenous group can be further divided into two categories, the neutral and the acidic. The neutral group contains the blood group substances. The acidic group can further be classified into sulfate-free and sulfate-containing. A reminder over here again that more the amount of sulfate present, more acidic is the heteropolysaccharide. So sulfate containing compound will also contain an amino sugar. So they have more acidic groups present in them. Let's talk in little detail about the sulfate free. Sulfate free can be further of two types. One is hyaluronic acid and the other one is chondroitin. You know that both these compounds are present in our skeletal network and in the extracellular matrix. We'll be talking about these compounds once we have a separate class on the polysaccharides. Now sulfate containing can be of different types. The first one is keratin sulfate. After that you have chondroitin sulfate, and the last one is heparin. Heparin is, as you know, is the natural anticoagulant. So this is the classification of heteropolysaccharides mainly into two categories: non-nitrogenous, nitrogenous. What is the basis of that? The presence of amino group. Non-nitrogenous, plant gum, mucilages, pectin and agar. We will not be talking in detail about these. And nitrogenous, the ones containing the amino group. They can be neutral or acidic. Neutral ones are the blood group substances. The acidic one further into two categories: sulfate free and sulfate containing. The sulfate, under sulfate free you have two: chondroitin and hyaluronic acid, and in sulfate containing you have keratin sulfate, chondroitin sulfate and heparin. The best thing for you to remember is to make this chart, draw it on a piece of paper and paste it on the wall next to you so that you can keep on revising, because it gets confusing. Now let us discuss about other sugar derivatives which have biomedical importance. Going over to glycosides, sugars form glycosides with other compounds and with each other. Glycosides are formed by condensation between the hydroxyl group of the anomeric carbon, the C1 of a monosaccharide, and a second compound that may or may not be a monosaccharide. When it is not a carbohydrate residue, it is known as a glycon. These glycosides are widely distributed in nature, and this aglycon or the non-carbohydrate residue can be an alcohol, a glycerol, a steroid compound, a phenol or adenine. For example, when adenine becomes a glycoside, it combines with glucose, so it becomes adenosine, and that is the compound that helps in the formation of ATP. Cardiac glycosides are also important in medicine. They contain steroids as aglycons. So that means cardiac glycosides contain steroids and a carbohydrate residue. These includes derivatives of digitalis and strophanthus, and quinone, which is an inhibitor of sodium and potassium ATPase in the cell membrane, especially in the cardiac membrane. Other glycoside includes antibiotics such as streptomycin. Now going ahead with deoxysugars. Deoxysugars are those sugars in which hydroxyl group is converted into hydrogen group with the removal of water. And these sugars are more stable and they are less prone to hydrolysis because of the absence of the unstable hydroxyl group. So deoxysugars are those in which hydroxyl group has been replaced by hydrogen, a lack of oxygen atom. An example is deoxyribose in DNA. It is a very important example, and because of this deoxy nature of the ribose, the DNA is more stable. Another example over here is deoxy sugar L-fucose, which occurs in glycoproteins. Over here I've shown you the structure of the deoxyribose sugar which is present in the DNA. Over here you can see that at OH position, where a hydroxyl group was supposed to be there, there is only presence of one hydrogen group. Next sugar that we should discuss is hexosamines. So when a hexose sugar combined with the amino group, it leads to formation of hexosamine. And the most common combination point is again at position 2 of the hexose sugar. So if it is glucose combining with the amino sugar, it will become glucosamine. If it is a galactose combining with the amino sugar, it will become galactosamine, and so on. So it contains an amino group components. So mainly these amino sugars. Remember, we have done the classification of heteropolysaccharides. They are the heteropolysaccharides which contain the amino group or the nitrogenous compounds. They are components of glycoproteins, gangliosides and glycosaminoglycans. The amino sugar include D-glucosamine, a constituent of hyaluronic acid, D-galactosamine or chondrosamine, a constituent of chondroitin, and D-menosamine. Several antibiotics like erythromycin are amino sugars, and they have, because of the presence of this amino sugar, they have very important antibiotic activity. So amino sugar acids, very important. Again, they are constituents of the glycoproteins and glycolipids. Neurominic acid. It's an aldol condensation product of pyruvic acid and D-menosamine. They're present in nature as acetyl derivatives with sialic acid residues, and they're also known as N-neurominic acid or NANA. They are present as constituents of nitrogenous oligosaccharides in milk. They are found in mucopolysaccharides and glycolipids like gangliosides. Muraminic acid is another amino acid sugar, structurally condensation product of glucose and lactic acid, and it is a constituent of the bacterial cell wall. So I have briefly revised with you all the important aspects of the classification of carbohydrates. Please go through it, and if you have any difficulties or any queries, please feel free to message me on my WhatsApp number, or you can email me on my email address. If you want to have one-to-one sessions with me, you are most welcome to message me, and it can be arranged. Thank you so much for listening to my lecture. Thank you.